Hi everyone, it's Brian. As you all know, I am a bit under the weather. You can probably tell by my voice. I've got two sets of slides here I want to get through. This osteology part two, and there's also muscle physiology. I'm going to do the best I can here. You will be responsible for this content for the final next week. I will be sending a review out for the final at some point later today, but we need to get through some of this material to get you prepared for the final. So we've already talked a lot about bones and structures and so forth, but we need to go a bit deeper into the sort of the workings of the bones. So of course, bone is tissue, which means it's made up of cells, and there are four basic cells in bone, the bone matrix. We have to remember that bone is connective tissue, and remember the definition for connective tissue are cells embedded within a matrix. So we do have that in bones, where we have the bone matrix and we have cells that exist within it. We have osteoprogenitor cells, which are osteogenic, that help prepare to become osteoblasts. Osteoblasts, if you remember, blasts means create. So osteoblasts create bone. Then we have osteocytes, which maintain bone. And we have something called osteoclasts, and its responsibility is to break down bone. And we can see them here in the bottom of the slide. The osteoprogenitor cell means that it can become an osteoblast and then eventually an osteocyte. So the osteoprogenitor cells themselves, they're unspecialized, and they grow out of mesenchyme. And remember, we've already talked about mesenchyme previously in clinical or in A&P1, and we know that connective tissue, all connective tissue derives from mesenchyme, and bone is a connective tissue. These osteoprogenitor cells can develop into osteoblasts. They can be found in the inner portion of the periosteum. They're also found in the endosteum and in the canals, which we'll learn about a bit later, that exist within bone that contain blood vessels. The second type are called osteoblasts. Now, these are the osteoprogenitor cells that do specialize to become these, and their whole responsibility is to create bone. So they don't divide. Once they're created, they just kind of hang around, and their whole purpose is to secrete collagen. So they help through the secretion of collagen to create the matrix. And you can see here in the picture on the right-hand side, here is an osteoblast that is creating matrix. It sits within and it's surrounding itself. We can see one here as well. The next type is called an osteocyte. So the blast creates the matrix by secreting collagen. Once that osteoblast is completely surrounded by bone tissue, it matures to form an osteocyte. So mature bone cells, they're called mature bone cells that were originally osteoblasts. It is the principal cell of bone tissue. Again, it has no mitotic potential at all. It just hangs around in its tiny little space surrounded by matrix. It's found on the surface of bone, and as I said, osteoblasts, once surrounded by bone matrix, this matrix that they're secreting, they become osteocytes. Osteocytes do not secrete matrix material. Osteoblasts create matrix material, not osteocytes. Osteoblasts form bone tissue, and osteocytes maintain cellular activities of bone tissue. So osteoclasts, their whole role is to help in bone resorption. And this is used in development, growth, maintenance, and repair. So during growth inside mom, we need some remodeling that needs to occur in some areas, and osteoclasts will help remodel. We also have to remember that bone is made up of calcium, and bone, we're going to discover, is actually deposited, or within bone, calcium is deposited in bone. And if we need calcium to maintain homeostatic levels in the blood, osteoclasts will remove calcium from bone tissue, maintaining the appropriate amount of calcium in the blood tissue. There are a couple of very good videos here to watch. I highly recommend watching them. So as I said, osteoblasts create the matrix. So our collagen within the matrix. So we say, because of that, we say that they create the matrix. So it contains abundant mineral salts. The matrix has lots of minerals in it. It is primarily made of a crystallized form of something called tricalcium phosphate, known as hydroxyapatite. Okay, so There also is some calcium carbonate. This is where you usually have a conversation around, that what you usually buy in the health food stores to maintain calcium is calcium carbonate, which actually comes from oyster shells. But the reality is one should be taking tricalcium phosphate, which is better absorbed and used by the body. So we have the osteoblast creating this matrix of collagen. Of course, collagen is soft, right? It's very soft. So the salts, once that matrix of collagen is created, we have deposition of crystallization of these hydroxyapatite crystals, and it crystallizes this soft matrix of collagen. So we say as these salts are deposited in the bone framework, crystallization occurs and the tissue hardens. This crystallizing of this soft matrix that's created by the osteoblasts is known as calcification or mineralization. Collagen fibers are necessary or the bone would be brittle. So in other words, if it was just crystals, the bone would be stiff and it would be brittle and it would break very easily. If you can imagine just moving around, walking, those sorts of things, the amount of stresses that are placed on bone tissue, a lot of forces go through bone, and if bone weren't able to bend even just a little bit, they would break. So this collagen fibers allows for a bone to have that small bending, which helps it deal with the kind of forces that are placed through it. And the fact that it can bend and be strong, this is known as tensile strength. Hi everyone, I'm sorry, I'm going to have to pause every now and then, blow my nose or cough or whatever, so if it seems a bit jittery, I apologize. So if we remember, we talked about compact bone and spongy bone at the beginning of the semester. So I want to take a deep dive into the architecture of compact bone. And we can see it here. And what I want you to take note of is there's little red dots in here, and those represent blood vessels that actually run through the bone. And we can also see here that there's configurations of circles everywhere we look. And these circles represent a certain part of bone. And these run up and down the length of the bone. So we're doing a cross section here, kind of a transverse section through a bone. And we can see these circles, and in the middle of each circle, you'll note that there's actually a bit of a blood vessel there. So that's what we're going to talk about. Compact bone, although it looks solid, when you look at a femur or something like that, and you saw, a bit of a cut one that I had from a cow that showed the medullary cavity, it looks pretty solid. Well, under this microscope, it is actually made of a concentric ring structure. And within this concentric ring structure, we have blood vessels, lymph vessels, and nerves that penetrate through perforating canals called Volkmann canals. So we can see here that we've got, these penetrating canals. Here is a circle here, and here we can see we've got this penetrating canal here. So here is the concentric circles here and here. And as I said, there's a blood vessel running up and down through the middle of the circle. Well, we can see there's a cross-sectional canal that sort of goes from one artery to the next that we can see here. That one that crosses crosswise, that penetrates through the canals or through the circles, is known as the Volkmann canals. They connect with the vessels and nerves of the medullary cavity and those of the central canals. So we can see here, we can see these circles, and we can see that we have a blood vessel that runs through the middle of the circles, and these are known as the central or the Haversian canals. And then, of course, we have these ones that cut across transversely, and these are your Volkmann's canals. So Volkmann's run across transversely, and your central canals run up and down, or your Haversian cells run up and down through the middle of these concentric circles. So we say the central canals run longitudinally through the bone. So you can imagine these circles and these Haversian canals run the full length of the diaphysis and all the way from one end to the other. So imagine a femur. You can see it running from one end to the other along the shaft, and they're fairly long. Around the canals, we find these concentric rings, and these concentric rings are called lamellae. So we can see here in the diagram a little bit closer, we have these circles that are here, and we can see that the circle, right, they're in almost like an onion, they run in these rings. Here's our central Haversian canal that we can see here, and here's our Volkmann's canals that are running here, and you can see the periosteum here. So Volkmann's here and Haversian canals here. We have these concentric rings, and each of these rings is known as a lamellae, right? So the lamellae are rings of hard matrix. So these rings have a central canal, and then they're like I said, they're like an onion, and there's layer upon layer of them, and these are known as lamellae, and they are actually that calcified or mineralized matrix that was originally collagen secreted by the osteoblast, and then was crystallized through the calcium. Between the lamellae, we have small spaces. So you can see here in this diagram, we see these large holes here. And these small spaces are called lacunae. Now remember I said that osteoblasts secrete matrix until they're completely surrounded by it. And once the osteoblast is surrounded by matrix, it then converts into an osteocyte. So as I said, the blast secretes the matrix all around itself, and once it's completely surrounded, it converts into an osteocyte. So the space where the osteocyte sits, which of course was originally the osteoblast, that space that the osteocyte sits in is called a lacunae. And you can see here again, you've got these dark areas here, these are all lacunae, and within these dark holes would sit the osteocyte. Now you'll note here that there are canals leading out from the lacunae and interconnecting and communicating with each other. We can see them running along here, heading one way or the other throughout the entire bone here. So here's our lacunae, and we can see these canals heading outward from them. These branches that head out, these small canals heading out from the lacunae, are called canaliculi. Think of canal, right? So radiating from the lacunae are very small canals called canaliculi, and they are filled with cellular fluid. Within these canaliculi and fluid, we find these long finger-like extensions or processes of the osteocytes. So the osteocytes sit in the lacunae and send out processes via the canaliculi, which then allows the lacunae to connect with each other via these canaliculi and the processes of the osteocytes. So we can say that there's connection between the osteocytes throughout bone. So we can say, therefore, there is a canal system throughout bone which allows nutrients and oxygen to reach the osteocytes. Each central canal, remember, central canal running here, each central canal and its surrounding lamellae, the layers of concentric hardened matrix, the osteocytes and canaliculi, of course the lacunae, right, with the osteocyte, and the canaliculi all together form what is called an osteon, also known as the Haversian system. So we can see here that this whole area here is considered the Haversian system or the osteon. We can see here where they've put it in brackets that each of these circles is an osteon or a Haversian system. Again, the central canal, the lamellae of the circles, the lacunae, the spaces, the osteocytes that sit within the lacunae, and the canals that branch out from them, the canaliculi, forms what is known as an osteon or the Haversian system. The areas between the osteons, of course, contain interstitial fluid. So we can kind of see a very thin slice of bone tissue here under a microscope, and you can see how they all communicate and interact with each other. You can kind of make out the rings here, the circular configuration here to some degree, and you can see these lacunae containing the osteocytes and these canaliculi then spreading outwards. Cancellous bone, of course, spongy bone we talked a little bit about as well, which are in the diaphysis, differs in microscopic structure as compared to compact bone. The first thing is, of course, that compact bone has the osteons, spongy bone does not. So spongy bone does not contain osteons. instead we find these needle-like spicules called trabeculae. So they're all made up, and you can see in the picture here all these spike-like or needle-like extensions, these spicules, and they're called trabeculae. We find the bone cells within the trabeculae. So we still have osteoblasts creating and osteocytes. Then once surrounded, they become it hardens. So within these trabeculae, we do find what was once osteoblasts becoming osteocytes. We do have them sort of embedded within here. So it's similar, except that, of course, the spongy bone has large air spaces within it. Nutrients are delivered and waste products are removed by diffusion through the tiny canaliculi that extend to the surface of the spicules. So we still have osteocytes within lacunae, and then we have these canaliculi, and they, instead of interconnecting, they move out into this open space. Now remember that the spongy bone would be completely filled up with red bone marrow. So this is what would allow, through diffusion, oxygen into the cells of the bone, of the spongy bone, and then of course byproducts out. Now when we look at it here, it seems like it's here, there, and everywhere. As I said, almost looks like sponge toffee. As I say, the placement of trabeculae seem unorganized and random at first glance, but in fact they're arranged along lines of stress, so their orientation will differ between individual bones according to the nature of the magnitude of the load. So this is a load map that we see here on the left that's been drawn, and these are all lines of force that run through the proximal femur here, for example. And these loads of force are represented here when we start looking at the pattern of the trabeculae within this proximal part of the femur. And we can see the linearness of it, and it's matching, to some degree, these lines of force. So the fact that they're lined along the force enhances the bone's strength immensely, and it's able to be very light but very strong at the same time. The arteries that supply the bone marrow in the medullary cavity provide the nutrients to the spongy bone, as I said, via these canaliculi that are these canals that connect with the marrow itself. Spongy bone, as I said, is light, and of course the trabeculae support and protect the red bone marrow that sits within it. We find this spongy bone and this red marrow in the hip bones, back bones, and the ends of long bones. And the fact that these are the only areas of red marrow in the adult, they are the only site of hemopoiesis, which is the creation of red blood cells in adults in these areas. The next thing we need to talk about is the development and growth of bone. So, of course, where did bone come from? And I've sort of spoken to a little bit in terms of this mesenchyme, which when we were talking about that earlier in A&P. The process of bone formation, which I've talked about a little bit here, is called ossification. So the ability to create bone is known as ossification. And when we're inside mom, our skeleton is actually not bone yet. It's actually connective tissue. So we say the skeleton of the human embryo is fibrous connective tissue. These membranes formed by embryonic connective tissue, so mesenchyme, and hyaline cartilage that are loosely shaped like bone. So we have these bone models that essentially arise out of mesenchyme. So we have mesenchyme that connects these osteoprogenitor cells, and then we have them, these osteoblasts, but they initially start out as cartilage. So we have fibrous connective tissue membranes formed by embryonic connective tissue mesenchyme and hyaline cartilage that are loosely shaped like bone. This model is a supporting structure for ossification. So it may not look like the bone it's eventually going to become, but there is a model that will eventually become ossified through ossification. Ossification begins around the 6th to 7th week of development. And that may surprise some of you that, when we think about when we first discover that you're pregnant, it's usually, a little bit on when you miss that first period. And by that point, you already have a bit of a nervous system, a cardiovascular system, and now you're starting to ossify bone. This ossification process continues throughout adulthood. So we have this constantly happening. Of course, as with children, we've got a lot of growing to do. We're quite small when we start out, and we get to be 6 feet, 7 feet tall. So what's happening there? But let's start first with development of bone when we're inside mom. So there are two ways to make bone. The first is called intramembranous ossification. This is the formation of bone directly on or within fibrous connective tissue. If we think of a newborn baby, they have soft spots on their head. These soft spots represent this fibrous connective tissue. So we say that they're known as fontanelles, so these soft spots. So these fontanelles are fibrous connective tissue, and eventually they become hardened bone by intramembranous ossification. I think, as we talked about before, these soft spots are required so that the head can take on, as it goes through the birth canal, can deal with the kind of pressures that happen while the child is being birthed. And then once out, then as time goes on, these soft spots become ossified or hardened. But they are this actual fibrous connective tissue that will eventually ossify and become hardened. So what happens? The first thing is we have these mesenchymal cells. Now the mesenchymal cells become vascularized. The whole key here to ossification is blood. So before this cartilage, so if we go back here to this slide, we say that the skeleton of the human embryo is fibrous connective tissue and hyaline cartilage. So they're loosely shaped like the bone, and they're soft tissue, hyaline cartilage, and connective tissue. Once we have an artery that pops into this soft tissue, then we can start to make bone. If we don't get blood, it remains cartilage, and that's going to be key for a little bit later on. So these mesenchymal cells become vascularized. They cluster and differentiate first into osteoprogenitor cells and then into osteoblasts. I already talked a little bit about osteoprogenitor cells just a moment ago. So the osteoblasts, right, osteoprogenitors that then differentiate into osteoblasts. So mesenchyme, they become vascularized, they cluster together, then they become osteoprogenitor cells, and then they become osteoblasts. Where all this is occurring, this cluster, this is known as a center of ossification. So we can see here that they're all starting to come together here. So we've got these osteogenic cells or osteoprogenitor cells. They're then going to convert. So we've got the mesenchymal cells here within this area. Then they're going to convert into these osteoprogenitors, which then are all going to occur into these osteoblasts. We can see it here within the cycle. Those osteoblasts will then start to create the matrix. They'll secrete the initial matrix that has yet to be ossified. Okay, so it's the collagen that's still soft. So they're starting to secrete the collagen here. And again, and as we know, as they become completely surrounded by their own matrix, they will then convert into an osteocyte. Okay, let's move on then. So that was this mesenchymal cells become vascularized, cluster, and then what you get is, of course, you get trabeculae occurring, just like we talked about, and then you get, it starts to form spongy bone initially, and then eventually you get compact bone on the outside edges and so forth, depending on what it's going to be. But the initial thing I want you to understand, it's happening within the membranes here of the skull, and that you get these mesenchymal cells to these osteoprogenitor or osteogenic cells, which then become osteoblasts, secreting the collagen, surrounding themselves. Once they're surrounded, the osteoblasts become osteocytes, and then that matrix starts getting impregnated with the calcium salts and starts to harden. The second type of ossification is called endochondral ossification. So if we remember, chondro means cartilage, so endo means within cartilage. So endochondral ossification means the formation of bone in hyaline cartilage. So in other words, the replacement of cartilage by bone. This does not lead to differences in structure of bone. The first stage is the migration of mesenchymal cells into the area of the bone formation. The cells increase in size and differentiate into osteoprogenitor cells. Now, as I said, the key is blood. So we have mesenchyme that has differentiated now into osteoprogenitor cells. If there is no blood in the area, those osteoprogenitor cells will become chondroblasts. So they're cells that are responsible for creating cartilage. Okay? If there's capillaries in the area, remember I said it's all about blood, then those osteoprogenitor cells will become osteoblasts. So where there's no blood, osteoprogenitors become chondroblasts, which creates cartilage. And where the capillaries are present, the osteoprogenitor cells become osteoblasts, and then you start creating the whole process of the matrix and so forth. Endochondral ossification is the most common way to make bone. So we can see here we've got the cartilage model here, and then we get the penetration of blood into the area via a capillary or an artery. Remember, we had the nutrient foramens in the long bones that we looked at when we were looking at the femur. Well, that nutrient foramen represents the first time that an artery penetrated into the cartilage model, and it stays in that bone until death. So all of a sudden now we have blood in the area. So those osteoprogenitor cells, of course, are going to become osteoblasts. They're going to secrete matrix until they're surrounded and become osteocytes. And then we're going to have those osteocytes maintaining the soft matrix, which eventually then will become calcified or ossified and become hardened. And we can see it starting from the center here and working outwards. Now, we know that the very ends of bones, on both ends, are made of hyaline cartilage. We get these proliferating chondrocytes, which are going to work up to these edges and will make the appropriate cartilage at the articular ends of the bone. And behind that will be bone formation. Of course, within the shaft, you're going to get compact bone made. And within the distal diaphyses, you're going to get spongy bone made. So again, though, the key point here is that these osteoblasts are there because we have blood that's come into the area. So we can see eventually we have this proliferation of bone here out the edges. So we have the compact bone here on the outer edges. And then we would eventually we'll get osteoclasts, of course, which will eat away and create the medullary cavity. And then we'll have bone marrow. In the case of a fetus, will remain red, eventually to become yellow in adults. And we have this proliferation of blood tissue throughout it. So, cartilage model. We've got mesenchyme here, which then differentiates into osteoprogenitor. The presence of blood, the osteoprogenitor cells become osteoblasts, which then surround here, start secreting the matrix. Out here, because there's no blood, these will convert into chondroblasts. Again, blood, osteoblasts, no blood, chondroblasts. The osteoblasts, of course, will secrete the matrix until they're fully surrounded, and they'll start to surround themselves, and then once surrounded, of course, become osteocytes. Then you'll get mineralization occurring here, and then further specialization, the removal of bone tissue here to create the medullary cavity with the hardened compact bone here on the outsides as it's moving outward from the middle towards both ends. We can see here that we've got chondrocytes remaining here, right, and have created the cartilage. But blood does go into the center, which will start the process of creating spongy bone here in the center. Outwards here will have no blood in it, so chondrocytes will create the articular cartilage, and in the center here we'll have blood, which will create spongy bone. So this slide will kind of formalize what I just talked about. So and mesenchymal cells crowd together to create a template of the future bone, right, creating that model. The mesenchymal cells will differentiate into chondroblasts that produce the hyaline cartilage initially, right, because remember this was initially made of nothing but hyaline cartilage. A membrane develops around this model, and this is known as the perichondrium. So because this is just cartilaginous tissue, there's a skin around it. It's known as the perichondrium. So mesenchyme differentiate into chondroblasts that initially produce the model. Okay, and then a skin forms around it, and this is known as the perichondrium. Okay, the perichondrium. Now we have the cartilage model, which is this. Okay, we have the cartilage model that will eventually become the future bone. This model will grow in length due to continual cell division of the chondrocytes. This growing of length is known as interstitial growth or growth within. It will also grow in width. So growth in thickness is due to production of new chondroblasts from the perichondrium. So length is due to cell division of chondroblasts. Width or thickness is due to production of new chondroblasts from the perichondrium. And this is known as appositional growth. So if we look here, we've got the chondroblasts that are dividing towards the ends. And then for width or thickness, this perichondrium starts producing new chondroblasts from the perichondrium inward, thus creating the extra thickness. So this extra thickness is known as appositional growth. Now, as I said before, what happens is we have a nutrient artery that enters the perichondrium and then through the bone, through a hole in the bone called the nutrient foramen, which we have noted. And this occurs basically in the mid-region when it's in mum. It does get to the ends as we grow taller, and that makes sense, right? Because the bone's getting longer. So what happens is, now that we have blood, we now will stimulate osteoprogenitor cells in the area of the blood to differentiate into osteoblasts. And these osteoblasts lay down a thin shell of compact bone called the periosteal collar. Once the perichondrium starts to form bone, it becomes the periosteum. So we can see here we've had blood that's entered in, and we end up with this ossification center. And because we have blood, they will start to produce bone tissue, which will form the collar around the midpoint here. Once that collar starts, then because we have the presence of blood, the osteoprogenitor cells will differentiate into osteoblasts, and the whole thing I've talked about so many times. The osteoblasts will surround themselves in matrix, and once they're surrounded in matrix, they'll become osteocytes, and that softened matrix then will start to ossify with the stricalcium calcium, and then it will start to harden and become the bone, and then of course we get the creation of the medullary cavity as well. So where the blood went in, and we now created the collar. In the middle of the model, capillaries of the periosteum grow into the cartilage along with the osteoblasts, osteoclasts, and red bone and red marrow cells. And this is known as the periosteal bud. This center area where this blood supply starts to begin in the center, this is known as the primary ossification center. And from this central area where the blood went in, everything starts to move out towards the ends, either end, either way. So we have, as this is moving outward, right, towards the epiphyses here and here, right, we start to get this modeling that's occurring, and we can see here that we have this primary ossification center. It looks better in here. We can see it here, and it's going to start moving outwards in either direction. Because we now have bone cells here, remember, the perichondrium has now become the periosteum. And we get the capillaries of the periosteum. They also grow into the cartilage, along with the initial blood vessel that went in through the nutrient foramen. And now we have differentiation into osteoblasts being created, of course, creating matrix. We have osteoclasts, which are maintaining, and so we have osteocytes, which are maintaining, and now we start to develop osteoclasts, because we need those to start remodeling, so we need to start creating that medullary cavity. And now we start getting the creation of red marrow cells, because now we have the space to put them in. And remember, inside mom, in long bones like the femur, the fetus does have red bone marrow, whereas in adults they have yellow bone marrow. So once we have this primary ossification center, we can now start developing the diaphysis and eventually the epiphyses. So of course, the diaphysis of the model is then completely replaced by compact bone. And remember, they're working outwards toward the epiphyses. Now, once we get out to the epiphyses, we need, because we're getting far enough away, we need a blood supply to start creating spongy bone in these epiphyses. So we do get arteries that enter the epiphyses, and this is where we get secondary ossification. So primary ossification occurred at the midpoint, which creates the diaphysis. And then we get blood that penetrates into the ends of the cartilage model, and we start getting blood there entering in, and we start getting chondroblasts being replaced with osteoblasts, and we start creating the spongy bone to make up the epiphysis. And this area is known as the secondary ossification center. Usually this starts occurring right around the time of birth. And there are some differences here. Of course, the first is that this is the cartilage is replaced with spongy bone, not compact bone. And of course, being the epiphysis, there is no medullary cavity. We just get it completely filled with spongy bone. The chondroblasts are retained over the articular portion at the very ends of these bones, of the epiphyses. We do get hyaline cartilage that is retained, and we also get hyaline cartilage that's retained near an area called the metaphysis. If you remember, the metaphysis is between the epiphysis and the diaphysis. And we do have hyaline cartilage remaining there for what is known as the growth plate, which we'll be talking about very shortly. So you can see here we have our primary ossification center here heading out towards the epiphyses. We then have the epiphyseal artery and vein that penetrates the epiphyses. And of course, we need blood to start creating bone tissue here in the epiphysis. And of course, that'll move outward towards the edges, but we will have chondroblasts that are remain out here on the edges that will maintain that there is cartilage here, of course, at the articular area of the bone and to form the joint itself. So the articular cartilage or the hyaline cartilage is maintained here. And as we can see, our hyaline cartilage is also maintained in this area right here in the metaphysis, which is where we find the growth plate, which is what we'll talk about now. So remember, appositional growth, is lengthening of bone. So appositional growth continues throughout childhood. Of course, remember, we have to go from a little baby to, 6 feet tall in some cases. And long bones lengthen via the diaphyseal side of the epiphyseal plate. Now let's think for a moment where that is, the diaphyseal side. So the diaphysis is the shaft. So that means that bones lengthen from the diaphysis towards the epiphysis. So we're saying that long bones lengthen via the diaphyseal side of the epiphyseal plate. Now we can see here in the diagram in pink, this represents the growth plate here, the epiphyseal plate. Down here is trabecular bone, and down here would be the epiphysis, and up here would be compact bone that would represent the diaphysis. So we have 4 zones here: resting, proliferating, hypertrophic, and ossification zones. So everything is moving in a net direction from here towards here. So the first part of the epiphyseal plate is called the zone of resting cartilage. This is near the epiphysis. It consists of scattered chondrocytes, and they do not function in bone growth. They anchor the plate to the epiphysis. So we can see here's the zone of resting cartilage right here. Sorry, I may have told you this upside down. I apologize. So this would be the resting zone here. We'll stay with this picture here though. Here's the zone of resting cartilage right here. Okay, and that's on the epiphyseal side, right here. I have a larger picture here. We can see this is the area of the epiphysis here. Here's the diaphysis down here. And we can see the zone of resting cartilage there. It's near the epiphysis. It is made of scattered chondrocytes. As I said, they don't function in bone growth. This is the anchor of the growth plate to the epiphysis. The next one away of it is called the zone of proliferation. So we can see this area right here. It's slightly large. It's made of slightly larger chondrocytes, but they're arranged like stacks of coins, and they replace each other at the diaphyseal surface. So we can see here is our zone of resting cartilage, and here is our zone of cartilage proliferation. So they're from the diaphyseal side toward the epiphyseal side. These chondrocytes are replacing each other. So we can see from the diaphyseal surface, from the diaphyseal surface here, they're replacing each other up towards the epiphysis. Zone of resting cartilage is right here, and we have the zone of proliferation moving this way. So everything's moving in this direction. Our next is called the zone of maturation. So of course, we start off with these immature chondrocytes. They're starting to mature, so they're getting larger as we move towards the epiphysis. We get lengthwise expansion of the epiphyseal plate because we get cell division of the chondrocytes here. Once they start to mature and divide out, they basically what we find is a zone of calcification. And they're only a few cells thick. They're mostly dead cells because they're surrounded by calcified matrix. And this allows for a good cement into the diaphysis. So we can see here in this diagram, here is our diaphysis. We have this zone of calcified cartilage, which you can think of as the anchor of the growth plate into the diaphysis. Then we have this zone of the creation of cartilage, so the zone of hypertrophic cartilage here, also known as the zone of proliferation. Then we get zone of proliferation. Then we get this zone of proliferating, sorry, good Lord, my brain's not working because I'm not well. The diaphysis is here. We have the zone of calcified cartilage here, which anchors it to the diaphysis. We have the zone of resting cartilage here that anchors the growth plate to the epiphysis. So calcified cartilage anchors to the diaphysis. Resting cartilage anchors the plate to the epiphysis. We then have the zone of proliferation, which is the creation of chondrocytes that we can see here. So chondrocytes are being created from resting cartilage, from the direction of resting cartilage towards the diaphysis here. And we have then them growing and dividing longer. So what ends up happening is we have it moving in this direction. So direction of ossifying of cartilage is moving this way, but we also have proliferating cartilage traveling this way. So ossification, we have the creation of chondrocytes here, and they're maturing towards the ossified bone. And as they divide and become ossified, we have net movement in this direction, elongating the bone. That ends osteology part 2. Make sure you check in with the book as well. It's all in there. The pages are in the syllabus. And I will be sending out an announcement in these once the second one gets done. Thanks so much.